The triplet code. What is a gene? How do genes code for polypeptides? So a gene is a section of DNA that codes for a particular polypeptide or protein. The code is the sequence of bases. Each gene has a specific locus or position on a chromosome. Genes determine the nature and development. So the DNA sequence determines the amino acid sequence, and proteins and enzymes are formed, and enzymes enable metabolic pathways, and metabolic pathways help determine nature and development. The triplet code must be a minimum of 3 bases coded for each amino acid because there are 20 amino acids that occur in proteins. Each amino acid must have its own code. There are 4 bases, A, G, C and T. If each base coded for 1 amino acid, only 4 amino acids could be produced. Using a pair of bases, only 16 different codes, which is still inadequate. 3 bases codes for 64, so there's more than enough for the 20 amino acids. As there's 64 codes and only 20 amino acids, some amino acids have more than 1 code. In eukaryotes, much of the DNA doesn't actually code for an amino acid. This is called introns and can occur in genes or as multiple repeats between genes. Polypeptides or proteins. The sequence of bases codes for the sequence of amino acids or primary structure. Each amino acid is coded for by 3 bases, a triplet. Not all DNA codes for polypeptides. Non-coding sections within a gene are called introns. Some sections between genes consist of multiple repeats. For example, tartar boxes. So you can see within a gene, so this is mRNA that has been transcribed, so this is known as pre-mRNA. But then what happens is the introns, the non-coding bits, are spliced out so that all you get left with is the exons, the bit that actually code. And that is your mature mRNA. So some facts about triplet codes. The code is degenerate, which means that most amino acids have more than 1 code. And the code is always read in one direction. The start of a sequence is always the same triplet code, and it codes for methionine. That is removed before the polypeptide is formed. 3 triplet codes don't code for amino acids. They are stop codons, which mark the end of polypeptide. The codes are non-overlapping, so each sequence is only read once. And the code or the base sequence is universal or the same in all organisms. So in summary, a sequence of 3 DNA bases called a triplet codes for a specific amino acid. The genetic code is universal or non-overlapping, and it is degenerate. In eukaryotes, much of the nuclear DNA does not code for polypeptides. For example, there are non-coding multiple repeats of base sequences between genes. Even within a gene, only some sequences called exons code for amino acid sequences. Within the gene, these exons are separated by 1 or more non-coding sequences called introns.